Tracy worked for more than 30 years at NOAA's Northwest Fisheries Science Center, ending up as the director of the Environmental Conservation Division, where his research portfolio included environmental toxicology and chemistry, assessing oil spill impacts, harmful algal blooms, seafood safety, and watershed processes. Following his retirement, he served as the science advisor for NOAA's Oceans and Human Health Initiative from 2010 through 2014, and was a technical advisor to NOAA and other natural resource trustees charged with assessing injuries to marine mammals and sea turtles after the Deepwater Horizon oil spill from 2010 to 2016. He continues to consult, this does not sound like a retirement, he continues to consult on environmental and human health risks. Dr. Collier received his PhD from the University of Washington in 1988, has over 160 scientific publications, and plans to retire again someday. A fun fact about him, he and his daughter raised 2 pigs and 6 turkeys for meat in 2016. The largest turkey dressed out at over 35 pounds, and a not so fun fact is that collecting turkeys is a pain. All right, so are we good to go, Tara? All right, we'll roll with it.
So should I use this?
Use one in your hand.
Okay. Well, thanks so much. I appreciate the invitation to come here. Yeah, after that introduction, my retirement didn't work out too well because of the oil spill, which happened 3 weeks after I left the lab, and I had worked on a lot of oil spills in my life. So when that oil spill happened, it did mess up my retirement plans. So what I want to point out is I'm giving this talk because other people are still working, not retired, and very, very busy. And those people are Brian Stacy and Karis Mitchellmore. Brian with NOAA and Karis with the University of Maryland Chesapeake Biological Station. I've liberally used their slides in putting this presentation together. Any mistakes, however, I've done some alterations. Any mistakes, or more likely any misinterpretations, are entirely on me, because I am even less of a turtle biologist, I think, than anybody in this room. My career was all toxicology, almost all fish, and then when the oil spill happened, I was asked to help out with the sea turtles and the marine mammals, which for me has been, it's been a fascinating experience, and I do know more about sea turtles than I did 7 years ago. So I wanted to quickly summarize my entire career in this one slide, which is the effects of how oil affects organisms. Basically, you've got in this case, a fish, because I did all my work on fish, but it could be a turtle or a dolphin. You've got direct effects. You've got narcosis. The fumes of the oil can basically put you to sleep, and if you're a sea turtle swimming or a marine mammal, that's not good if you have to breathe air. What we call receptor-based effects are those things that affect an animal through the compounds in the oil, actually interacting with the animal. In this case, some PAHs, and those are toxic components of oil, what we call the aromatic hydrocarbons. Some can cause cancer. I will just say cancer is not a generally very common endpoint with oil spills, because the types of PAHs in oil are generally not carcinogenic. And I throw that in here because I saw some of the questions from the audience about the relationships between the oil spill and some of the papillomas that are being seen in turtles. There could be a linkage, but it is probably not a direct linkage of the carcinogenicity of the oil itself. Edema means the accumulation of fluid, things that happen when your circulation doesn't work well. Cardiovascular dysfunction, effects on the heart. And then certainly in the case of sea turtles, physical coating, the effects on behavior and the ability to move, and how well they can breathe, and if they ingest a lot of oil, what it does inside their GI tract. Indirect effects are things like reductions in the food source, impacts to the habitat, and certainly those are of great concern for oil spills. Down here I've talked about what we call sublethal and delayed effects. I hate this word sublethal, because everybody says, so you didn't kill them. So why do we care, especially when we're doing damage assessment where we want to know about mortality. But sublethal effects mean you didn't kill them right now. But you may have done things like you may have affected DNA damage, you may have affected their adrenal system, their ability to respond to stress, their blood chemistry, they could get dehydrated, have behavioral changes. These things did a number of studies that are very difficult to do in feral, free-living animals. are linked to a number of things, including immune function, the ability to resist disease. So this is where you could have a link between some of the papillomas and a stress like an oil spill. Bioenergetics, their ability to actually, like Nathan was just saying, they've got to use their yolk sac as a little battery. If they're not able to efficiently use energy from the yolk, they're not going to get as far. Decreased survival, growth, and reproduction. All of these things, especially down at the bottom, the disease resistance down to decreased survival and reproduction, these are things that make up a population's viability. If you're deficient in these, you may not be dead, acutely dead, but if you go out into the real world and you can't swim as fast or you can't resist disease, what we'd say is that you are, even though we call this the sublethal effect, you are ecologically dead. So it is an issue. If anybody can ever come up with a better word than sublethal that I can use in these talks, I will be eternally grateful. And then it'll probably be time for me to retire. So just real quickly, Nathan, I think, gave you a real good overview. I want to do a little bit on sea turtles in the Gulf of Mexico 101. Me, a toxicologist working on fish my whole career, talking to you guys about turtles is probably, let's not spend too much time on this slide. But basically, the main thing is that we've got this oceanic phase, the small turtles that Nathan was talking about in the modeling. We've got the neritic phase, or the nearer shore phase, which are larger turtles. And then we got the terrestrial turtles and the nesting beaches. And I'm going to focus mainly on these first two, on the oceanic and the neritic. There's people here, Donna Shaver, Chris Schmidt, who have done a lot of work on the terrestrial side. And if you have questions about the nesting turtles and the hatching success and all that, you should probably talk to them. So this is how we look at the issue of sort of what we call a damage assessment. We think about the issue of exposure. How do we measure the exposure of the turtles to the oil? Then we have this really messy box in the middle called injury determination. How do we actually determine what the injuries were of the turtles? And then we take all this messy stuff in the middle and we turn it into an injury quantification, and we try to get total exposures and injuries, and we expand it to the entire spill footprint, much like Nathan was talking about through his modeling. And we then try to come up with the total number of sea turtles lost. And for the most part, where we really have good information is in this exposure, the physical exposure of the oceanic turtles, and then in the response, the exposure of the nesting turtles and the relocation of nests. That's where we really have a lot of good information. I'm going to talk some about these things in the middle and how we use that to basically expand our injury determination for the damage assessment. So again, Nathan gave a good introduction to this issue of the directed capture. This is where we probably have the best indication of exposure of the oceanic turtles following the spill, where it was not a random sampling. They went to where they expected the turtles to be, in convergence zones where you had rack coalescing, and that's where you found the turtles. The point was to try to get the turtles out of the area, get them into rehab, either before the oil got there or once they were oiled, take the oil turtles into rehab and keep them alive so you could eventually release them. So that was really, it made it hard to then extrapolate that into the full Gulf, because again, it was not a random survey. But again, Nathan gave a good, I think, we all agree on the numbers, so we have nothing else to worry about. So I'm glad that he was able to put behavior in and come up with the right number. That's my attitude anyway. But anyway, you could use this for, they also did surveillance for stranded turtles on the beaches. And on the right side, I'm not going to go into the chemistry, but this is showing indications of oil, in this case in the enteric contents in the gut of a turtle. This is the oil itself. This is what oil from the gut looks like. And you can see there's a really good match showing that this turtle that had oil in its gut probably came from the Condo Formation. And then if you look in the liver in this case, or other matrices in the animal, the fingerprint's not the same. And that's one of the things that makes it really difficult to determine exposure of turtles or other animals, other vertebrates to oil. Anything with a backbone basically metabolizes oil really efficiently. So what you see in the tissues is not the same as what you see in the oil, even though this is what the animal was eating and ingesting. And so this shows different degrees of oiling of the turtles during the directed capture, from minimal to heavy. And I'm going to come back to that when I talk about our toxicology estimation. So in terms of looking at exposure, we had the directed capture as a direct determination of exposure. We were getting exposed oiled turtles. We did the surveillance for the stranded. And then the other thing we could do was to look through aerial surveys, try to look at, this was for larger turtles. Again, as Nathan said, you can't see the little oceanic phase, but the larger neritic turtles, you can see through aerial or sometimes vessel surveys. And you can imprint those visualizations of these turtles over this, again, this cumulative oil footprint with a level of intensity. And then you can count how many turtles you saw in these aerial surveys. And these are done much more with a structured, statistically robust design. So you can take these aerial surveys, which you still have a real problem with detection, especially as turtles get smaller. But you can then use that to extrapolate to a full Gulf estimation of the number of turtles that got into the oil. So in terms of doing the injury, the directed capture turtles, they did a veterinary evaluation of the turtles when they brought them into rehab. They did a lot of blood chemistry. Those few that did die, they did a necropsy. They took samples for some of those internal measures of oil exposure that I showed you. And then there was a laboratory exposure study using a surrogate species, red-eared sliders and snapping turtles that was conducted. I'm not going to talk about that, but again, during the discussion, I can bring that up. I will just say the laboratory study was shorter term than the exposure of the animals in the Gulf, and it gave indications of injury, but not as many indications as we anticipated when we started the study. But there were indications of injury from that. And then this last one is this toxicological estimation based on estimated dosage and published toxicity data, and I'll talk about that a little more. And then again, the nesting turtles, there was this whole issue with the nesting beaches and relocation of beaches. Those eggs that were relocated, again, if these animals return to their natal beaches, but you've moved them to the other side of Florida, they're expected to not do as well because now they're displaced. And again, for the purposes of injury estimation, those were considered to be lost turtles, even though the eggs were moved. In terms of quantification, again, I'm not going to go through the details. Nathan did a great deal explanation on the modeling as to how it comes up. And again, it's very similar where you have your different turtles, your what we call pelagic, or same word, oceanic life stage, the smaller turtles, the neritic turtles, and you have the severity of the oiling over time. And you basically take and extrapolate as best you can all that information on oiling of turtles throughout the Gulf, and you put it together to get an estimation of how many, initially the estimation was how many turtles, especially in the oceanic phase, were heavily oiled. That really gloppy one that everybody uses. I used it on the title slide. Nathan had one. There was one of the heavily oiled turtles. We basically assumed every animal that had that degree of heavy oiling was going to die if it wasn't rescued and rehabbed. So when we extrapolate to the number of heavily oiled turtles, those are all considered to be mortality. And again, the reason is it's this physical impact. These animals are mired in oil, they can't really move, it's sunny, it's very hot. When Brian Stacy took some oil temperature readings with turtles in the middle, it was well in excess of 130 degrees Fahrenheit. These animals are not going to survive in that environment without any intervention. And then again, the toxicological estimate is what I'll be coming back to. Oh, I see. I went back one slide. So I was talking about the wrong slide. But anyway, you guys were watching what I was doing. So the issue is that we get for the heavily oiled turtles, those are considered to be all dead. And then there's an upper bound for less than heavily oiled turtles. What do we do about those where we know toxicologically oil is toxic to vertebrate species, but we don't have direct evidence of dead turtles from those lesser degrees of oiling? So simplifying how we look at injury for the Deepwater Horizon NRDA, its exposure equates to injury, and then we try to figure out how to turn that into damages. Again, the veterinary-based assessments were those turtles picked up during the directed capture, brought into rehab, cleaned up, fed, and then released after they appeared to be returned to health. However, the veterinary-based assessments concluded that based on the clinical data and the indications of clinical aberrations in those turtles, that 25% of those turtles would have died if they had not been rescued and rehabbed. So there was this bound of indications of what the vets call derangement, which I think is a great word, but it just basically means they're not normal. So the abnormalities in the clinical indications in these turtles in veterinary estimations would have amounted to 25% mortality. And then again, I'm going to spend a little more time at the end here talking about this issue. Talking about this issue of how do we take the toxicology-based assessments and the comparative toxicology, and how do we use that to better estimate how many sea turtles died as a result of the Deepwater Horizon spill. So again, here's the 4 levels of oiling. Here's a minimally oiled turtle, moderate, minimal, small, moderate, heavy. Anyway, categories 1 through 4, okay? And 0 would be a turtle with no oiling whatsoever. And so the numbers of these turtles that were having these degrees of oiling during the directed capture were recorded. And what was really nice was Brian and his coworkers noticed that when they were capturing these turtles, a large number of them had oil in their oral cavities. They were ingesting oil. And turtles, again, you guys know turtles better than I do, but the heavily structured esophagus, when the oil gets into a turtle's mouth and throat, it's not going to come out very easily. So we were able to they measured the number of turtles of each of these categories that had ingested oil, and they also estimated the amount of oil in the oral cavity. And so with that information, we could actually then estimate what was the oral dose of oil per day in these turtles in these different oiling categories. And I'm showing this slide because basically our numbers really derive about this middle number, the mean oral estimate. But we also looked at the minimum, and we looked at the maximum, and we put a range on both. And so we have tons of numbers, but the numbers that really count for most of the calculations are these bold numbers right here. But again, being scientists, we like to show that we've considered all issues of uncertainty and variability, because if we ever go to court on this, we want to show that we weren't so narrow-minded as to really focus on just this, but really we just focused on that. So we wanted to compare these. Then the next steps were to take this information on an oral dose, compare this with studies that have looked at toxicological effects of oil in turtles and other vertebrate species. There's actually very, very little done in turtles, almost nothing. There's been some, but it was intriguing. But the real good information actually comes from mink and other laboratory studies. But take that information and then use that to provide estimates of mortality for these oiling categories 0 through 3, the minimal, light, and moderately oiled. Again, heavily oiled turtles, the assumption is that they have died as a result of physical impacts of the oiling. And then use that to augment what we saw in the direct capture observations and get an estimation of mortality for all juvenile oceanic sea turtles in the impact zone. So what we get is, again, the heavily oiled turtles are considered 100% mortality. Of the ones that were moderately oiled, we get 85% mortality because we estimated that their daily dose levels far exceeds a toxicological endpoint that would give them a failure of their adrenal system, render them unable to respond to stress, and basically they would die in short order. And we figured that 90 to 100% of those turtles that ingested oil would be at risk of dying from adrenal insufficiency, but only 93% of the turtles in that had ingested oil. So we take the 90% times the 93% and we get an 85% mortality estimate for turtles in category 3. Turtles in category 2, we figure that the mortality due to this adrenal impact is going to be in the range of 65 to 80%, but again, only 76% of these turtles had ingested oil, so if you multiply those two together, you get a 50% mortality in that category. In the lightly oiled or minimally oiled, you get basically the estimate of 50% mortality from the amount they were ingesting. But again, only 50% of the turtles in that category had actually ingested oil, so we come up with an overall mortality rate of 25%. And again, in those turtles that did not show any evidence of external oiling, they did not find any evidence of ingestion of oil, so we're assuming zero exposure and zero mortality in the unoiled turtles. Oops, sorry about that. So we published a paper that came out just about 2 months ago now on this toxicological estimation of mortality in oceanic sea turtles that were oiled during the Deepwater Horizon. Again, the reason was we were assuming that heavily oiled turtles were dead turtles, but we had many, many turtles that were only partially oiled or moderately oiled. And there's a lot more that goes into the overall calculations, including the time of year as to when the turtles were captured, what was the probability of oil still remaining in the area. There's a lot behind this, but overall the toxicological estimation concluded that 30% of all the oceanic sea turtles that were not heavily oiled, and so this is going to include, this is a combination of the non-oiled turtles in categories 1 through 3. 30% of all the turtles that did not get heavily oiled died as a result of exposure to Deepwater Horizon oil. Because the trustees came to me as sort of the tox lead on this and said, can you give us any estimate of toxicology? And a lot of my toxicology friends said, you can't possibly do that. And I'm like, sure I can. I've got exposure, I've got oral exposure doses, and I've got laboratory studies. I can do anything with that. But smartly, I put Karis first as the author so that she can take all the real heat. But it actually was, in my view, it was an appropriate application of the expertise we have to give our best estimate of mortality, as I'll come back to. And I don't think we were being overly aggressive in estimating mortality. So this is the total number now. So this is from what we call the PDARP, the Provisional Damage Assessment and Restoration. What does P stand for at the end? Project, program, something like that. Anyway, there's a giant document online. You can look this all up. But this is a summary of the mortalities estimated from the oil spill. Basically, we have this oceanic, the surface pelagic juveniles, Kemps, loggerheads, green, hawksbill, unidentified, and total. And you can see these are the estimates of losses from the oceanic phase, losses from the this is larger juveniles and adults in the neritic phase, the hatchlings, and then the hatchlings again. Again, Donna and others can talk about the hatchling relocation, the relocation of nests, but a lot of those mortalities are coming from relocated nests that were assumed to not be contributing back to their original population. So those are the total numbers. Again, as Nathan just showed, there was good concordance between these numbers, although this number is not as high as the number he showed for Kemp's. These don't add up quite the same, so I'm sure he and I can talk about that later. And I just want to end with some concluding thoughts about the sea turtles and the Deepwater Horizon oil spill. Number one, in my opinion, these are likely low estimates. There's potential for additional effects from other exposure routes, especially inhalation, which we think in the marine mammal situation, the bottlenose dolphins in Barataria Bay, inhalation or aspiration of oil from the surface cause a lot of lung injury, we think. Well, we know the lung injury is there. We're still trying to figure out the route of exposure. But it's very likely that sea turtles, especially these oceanic ones, living right at the seawater interface, breathing the air right there, they will probably be aspirating some oil into their lungs, causing severe lung injury. There's unknown potential for effects, as I talked about these sublethal effects. There can be delayed effects that may manifest at either individual or population level for years to come. We really do, though, need to do this better. We need a better understanding of what oil does to turtles. I'll digress just 1 second here. We had an opportunity during the spill where there were some people doing studies on loggerhead turtles, on hatching and gender determination by temperature. Their permit required that once they hatched these turtle eggs out, the turtles had to all be euthanized. And I said, well, we've got these sea turtle babies. We could expose them to oil and see what it does to them. We're going to kill them anyway. But there was, it was a legitimate worry on the part of some of the trustees. We're trying to say to BP, you've killed our sea turtles, and look at our information of how we killed sea turtles using your oil. It's not a good PR, but from the standpoint of trying to understand the science better, we need to be able to make those leaps to do those sorts of studies. Anyway, I won't editorialize on that anymore. So we need to know about inhalation and aspiration of oil. We have very little information on what that could do to turtles. The chronic, sublethal, and delayed effects. All these things are real. We see them in many other species. We need to get a handle on how to do that. If we have to use surrogate species like freshwater turtles, that's fine. And again, I haven't really talked here about dispersants or dispersed oil. That is one of the first questions I always get. I'm not going to say anything about it because we know very little about it in turtles, but I bet when we have our panel discussion, you can ask me about dispersants and what the hell was in them anyway. The final slide, this is Brian Stacy's acknowledgement slide of the set I borrowed from him. Karis Mitchellmore has another acknowledgement slide that's just as long. I'm up here talking and doing the best I can to represent the turtle toxicology for you folks, but there are a whole host of people that have been working on this for many years. And they are all giving their all on this. And to me, this oil spill, as tragic as it was and the loss of human life, as tragic as it was for the environmental impacts, we have made huge strides in our understanding of oil and larger vertebrates like turtles and dolphins. Towards that end, hopefully we can keep that knowledge and use it for the next spill. So with that, I'll stop and thank you very much.